We'll start now. Hello, welcome everybody. I'd like to welcome you to this webinar, Gene Drive, a game-changing technology for grey squirrel control, jointly hosted by CONFOR, the CLA and the European Squirrel Initiative. Can I just go through some housekeeping items before we start the webinar in full? Please, can you mute your microphone and turn off your camera throughout the webinar? At the end of the event, we will run a question and answer session. To ask a question, please enter it into the chat function, unless you've already submitted a question. If selected, we may ask you to temporarily activate your audio and video so you can ask your question. We will try to get through as many questions as possible in the time allowed, but we are recording this session and we will try and save the chat file so we can see all your questions. The chat file will be looked at and we will be endeavour to answer everything that we can after the webinar if it hasn't been answered within the webinar, but we will also be running an article in the next edition of CONFOR's Forestry and Timber News that will further address much of what you hear today. Fiona, please, could we have the agenda up? While Fiona does that, we are absolutely thrilled to have two excellent speakers for you today. So Graham Taylor, many of you will know, Graham Taylor MBE, who is the chairman of the European Squirrel Initiative and managing director of Prior and Rickett Silviculture. He will be followed by Bruce Whitelaw, professor of animal biotechnology and interim director of the Roslin Institute. So CONFOR, I see lots of CONFOR members on call. We are the trade association, membership association for the forestry and timber sector from seed to saw. We are all too cognizant of the significant threat that grey squirrels pose to meeting our current and future woodland creation and management targets, net zero ambitions and nature recovery through the impacts grey squirrels have on the environment and our native red squirrel population. So alongside the suite of existing control measures that exist for controlling grey squirrels, CONFOR and its board have agreed that gene editing could be a real game changer. But before we start the webinar, here's a few words from our co-host, Graham Clark, who is Senior Land Use Policy Advisor for the CLA, advising members on forestry, woodlands and energy. Pass over to you, Graham.
Thanks, Caroline. Yeah, as I say, as Caroline said, we're very pleased to be co-hosting this with CONFOR and the European Squirrel Initiative. And many CLA members have woodland and want to plant more. And they're, as Caroline intimated, very aware of the damage that grey squirrels can cause to broadleaf woodlands in particular, and the effects on our native red squirrels. So they're very interested in any means to effectively control grey squirrel populations. And today we're going to hear about a really exciting new technique that could be used alongside various other methods that we use already. And when CLA's Forestry Committee discussed this issue a few months ago, they felt that it really did show great potential. So without taking up too much more time, let's get into the speakers. And it gives me great pleasure to introduce Graham Taylor, who's going to be our first speaker, and he's going to cover the grey squirrel problem and how we're currently addressing it. So, Graham.
Thank you. I hope you can all hear me. Let me just share my screen with you, and I'll go through my presentation. Hold on a moment. I can't talk and control a computer at the same time. There we go. Let's go back to the start. Right, can you just confirm that you can hear me and you can see that? Hopefully you can. Well, good afternoon, ladies and gentlemen. My name is Graham Taylor. As introduced, I'm chairman of ESI, and I'd like to thank you for attending the webinar today. I'm encouraged that there's over 100 owners, managers and agents who've tuned in to this presentation. And I think for many of us, grey squirrels are like that really irritating itch that never seems to go away, a dripping tap of disappointment. ESI's sole aim is ultimately to stop that itch, turn off the tap, and let our native wildlife and broadleaf trees flourish for future generations. I joined ESI at its inception in 2002. Oh, hold on, my screen share's gone. Was that going through? Can someone give me a nod? Bruce, was that coming through okay? No, oh dear, hold on. Share screen again. Is that now visible? Brilliant.
Yes, it is, Graham.
Sorry about that. I'll need another minute or two. Afternoon, sorry, ladies and gentlemen. My name is Graham Taylor. I'm chairman of ESI. I just want to thank the 100 or so landowners and foresters and agents who've joined us today. For many of us, I think grey squirrel are like that kind of really irritating itch, that dripping tap of disappointment that fails to go away. And ESI's sole aim really is ultimately to help stop that itch and turn off the tap. I joined ESI back in 2002 out of sheer frustration at the lack of impetus generally that was going on the anybody doing anything on grey squirrel control and the effects that they were having on young woodland. And I was in a room with some of those forestry greats, Bede Howell, Peter Goodwin were there. And for myself, it was the irritation of one's work just sometimes being utterly destroyed by the grey pest. I mean, not just the small amount of damage, it was the wholesale stand damage, irrespective of the efforts of control that were going on. And that has driven me to support both ESI and engage in red squirrel conservation in those areas, those woodlands and properties that we manage as a business across England, Wales, and now Scotland. So my day job is running a forest management company. Slowly but surely, we're becoming a company with national reach, and most of my time has been spent managing a lot of broadleaf woodland, particularly in the Welsh borders. We have some of the best oak growing conditions in the country, but this was just a continued repeat stuck record, as it were, despite good levels of control, still getting lots of damage. The problem we've got locally is that there's a correlation between fast broadleaf growth and damage, and unfortunately the Herefordshire soils are very, very fertile and broadleaves grow very fast. So we kind of have an even more difficult problem, as is experienced in southwest England as well than some other parts of the country.
Graham, just before you go on, you are on presenter slides, so we can see the text as well.
Oh, okay, hold on.
Fine. It's absolutely fine, but if you could get it onto full screen, that would be good.
I'll put you on full screen, sorry. Let me just hold on a minute. Pause, new share. Screen one. Is that right? Is that going to work? Hold on.
No, try again. These are the important slides anyway.
I don't mind people seeing my notes. As long as people can see, yeah, as long as I can see my notes. I put this slide up because really the damage is widespread. I mean, some people say how bad is this damage? It is widespread. It affects public forests. It affects private forests. It affects all these species, and slowly but surely we're going into a kind of world where actually the world is dominated by cherry, alder, and lime on the broadleaf front, and the conifers will continue to thrive. And if you look back at the big impetuses of planting over the last 30 years, we've got, think of the Southwest Forest, a 1990s initiative to plant. It was about 3,000 hectares of woodland planted, 2,000 hectares of broadleaves, 1,000 hectares of conifers. And a report recently just showed that in that 2,000 hectares of broadleaf woodland, there wasn't a kind of single oak tree worthy of note. They'd all been damaged by grey squirrels. The National Forest, we're now 25 years on to the National Forest. Some of the early adopters are seeing quite significant amounts of grey squirrel. National Forest Company trying really hard to help and support control mechanisms, but it's really difficult. There's a lot of damage. And I spent a lot of Forestry Commission research, did some work in the Welsh borders in the 90s, again, looking at publicly funded restocks, found that over 90% of the broadleaves were being damaged. The replanting after the 1987 storm, lots of people in southern England, same stuck record. And I just thought I'd show you'll all be familiar with the damage. Let me just show some images and I'll try and tweak this. balance of this screen. So it's more screen and less notes. There we go. Okay, so this will be the Chilterns. This stand was felled or blew down in the 1990 storm, replanted in 1991, 25 years on. It's got a flat top. It's not even apical dominance. That's quite consistent theme in the Chilterns. On the left there, that image is of a pause restoration site in the Wyre Forest. That's a 200-acre outlier. The Forestry Commission there felled 20 odd years ago some conifer, planted broadleaves at wide spacing. It's all like that. I mean, we're not just talking individual trees. It is all like that. Right-hand side, 2,000-acre estate in the East Midlands. We've only just got involved of late. And there's 20 years of restocks that all look like that on the right-hand side. Apical dominance disappeared. This image here is 500-hectare ownership in Berkshire. Same old story, affecting all their oak, birch, beech and sweet chestnut. And in that kind of context, I think sometimes I take woodland officers around, sometimes they're members of the public, and you kind of get used to what bad looks like and think it's normal. It's not normal. So I thought I'd just kind of encourage us with actually what you can achieve if you're absolutely top of your game with squirrel control. This is what normal looks like. Apical dominant, trees going up, forming that first length that becomes the valuable piece in 80 to 100 years time. So we've got some beech there on the left-hand side in Yorkshire, some sweet chestnut in Wales, 25-year-old oak in Herefordshire. Right-hand side is a mix of oak, beech and sycamore in the Cotswolds on a 3,000-acre estate. Left-hand side there is again a mixed woodland beech, ash and cherry. Again, the beech are going up, straight up to the sky. And on the right-hand side there, we've got a 25-year-old oak tree. The right-hand side image is a little bit of a false image. It's an image of a French oak wood that I took about 3 or 4 years ago. But in France, they're all like that. All the woodlands that you go into, which are being managed, are showing trees of that form and that quality. Our earlier woodlands are normal here. The damaged ones is normal there. In France, what is in the image there on the right is normal. So we're familiar with the costs. During lockdown, I sat and did a desk exercise myself based on forest inventory information, and I reckoned it was costing us about £40 million a year across the UK. And interestingly, the RFS and the UK Squirrel Accord had commissioned a study to do the same and come out with the same figure. So I'm probably about right. And what's interesting, of course, is that these figures are cumulative. It happens every year, somewhere else in the country. Every year, somewhere else gets damaged. And other stands get damaged, and they continue to get damaged. So actually, we're looking at 2.4 billion if it's been happening for the last 60 years. And because we continue to fell broadleaf trees from the Victorian area, which grew in the area before grey squirrels were here and or doing some damage, it will be in another 60 years, we'll have got through a whole oak rotation, and we will have to continue to import more because we have failed now and continue to fail now. And I reckon that figure is about £110 million a year. You might say, that's a lot of money. We import £14 billion worth of hardwood timber every year. That is not a lot of money. But it will be that big then. These are at today's prices. Property damage, British insurers talk about £20 million a year. Losses for carbon and biodiversity. I mean, those are very, very difficult numbers to quantify, particularly on the biodiversity front. I mean, what price do you put on the impact on wild bird populations? What price do you put on ancient woodland that's not being restored because owners are reticent to plant broadleaves because they're not confident about putting particularly oak and the other susceptible species back, so they continue to grow conifers. How to work out carbon, the impact, certainly that image in the Chilterns was pretty, that's not sequestering much carbon. So what have ESI been doing about it over the last 20 years? Well, we've been helping inform and shape the IMC work on immunocontraceptive. We've done lots of things with trapping technologies and trying to help people. So we were the first people talking to the good nature trap people. We've worked with other trapping technologies. We've got partners across the EU. We've done work on other things. I'm running out of time. But we've held conferences, seminars, looked at pine marten, fertility control. We've done lots of publications. We established RSST, and so it goes on. But fundamentally, we've been looking for this long-term solution. We will continue to hunt. It might take another generation of foresters to get there. But we think finally what's on offer, or the potential and the prospect with working with Roslyn, and the more we've talked to the professor, the more we're confident about the merits and the possibilities with it. That's why we're behind this thing. And I'll let now Bruce take us on. Thank you.
Thank you, Graham. Fiona, if you could share my slides, that'd be excellent. You know, for someone who spends his life sitting in a lab in an institute, it's really quite sobering to see both the scale, but also the range of impact that's happening in the forest there. It actually gives me even more conviction that I think we're on the right track. Anyway, who am I? My name's Bruce Whitelaw. I'm Professor of Animal Biotechnology at the Roslin Institute. I'm currently the interim director of the Institute, and we're part of the University of Edinburgh. And I've spent my whole academic career trying to develop and deploy genome or genetic engineering technologies, and I'll describe that in a few slides' time. If we could go to the next slide, please, Fiona. So I'm sure you're all well versed with the beauty of Edinburgh, and maybe even some of you have ventured onto the Easterbush campus and seen the new Roslin Institute shown in the slide here. We're famous for one specific white woolly sheep called Dolly, and we are currently doing a whole lot of activities to celebrate what would have been her 25th birthday. The shocking bit of news about that is I'm recruiting students into my group which are younger than Dolly. What's happened since? Well, we've continued. In fact, one of the drivers for Dolly was genetic engineering. We've continued to look for ways of using genetic engineering, and the current very exciting technology based on a number of reagents, but grouped together in what's called genome editing. And the pigs in this slide here show you what is perhaps our lead project. And we've generated pigs which are resistant to the PRRS virus, for those of you who are involved in the pig industry. International major burden on the pork industry. This virus, there are vaccines, but they're not fully effective. We have, by using genetic engineering tools, created animals which are resistant, and we're currently working with genus PIC to take them through the regulatory system in the US and deploy them around the world, hopefully in 4 or so years' time. That's what I do as my day job. And as part of this, I look to how we can use tools that we have for genetic engineering. And what I'm going to describe to you today is one called Gene Drive. Now, I appreciate this is probably not familiar territory for many of you. In fact, last time you probably did genetics was when you were at school. I apologize for those of you who've got a genetics degree and will correct me, but I will try and explain to you what Gene Drive is. Gene Drive is a use of genetic engineering tools, both GMO and genome editing tools. The next slide, please. This is my team, comprising of a series of academics with different skills, some in the lab, some molecular modeling, and we've got Shan there helping us with the business thing. This team's been in place now for perhaps a year and a half. We made some progress, and I'll explain that to you. But we are, the punchline is, we are not at the minute making squirrels. This is what we aspire to do. The next slide, please. So the project is called DIGBY, and it stands for Directed Inheritance of Gender Bias. And we proposed this in a paper a few years back, and it utilizes gene drive to, or looks at ways of using gene drive to control population size. Since we proposed it, gene drive has been shown to be feasible in mice, and a team in the States published this work, and the Australians have latched onto this as a way of controlling rodent species. You've all seen some of the imagery coming out of Australia with regard to the massive rodents, and what's significant there is that it is NGOs who you might not expect to be supportive of biotech who are coming out in support of this technology. So what is gene drive? Next slide, please. So this is my crash course in genetics for you. If you look at the lower part of this slide with the insects, and I'll explain why insects in a minute. What normally happens in inheritance is that a male and a female mate, and we each, whether you're a male or a female, carry 2 copies of the same gene. But you only pass one of those on to your offspring so that your offspring have one from mum, one from dad. And unless there's a selection pressure on that, that's what happens. Half the offspring carry the gene of interest, as shown on the left-hand part of this slide. Gene drive drives that gene, that specific genetic marker, through the population to a point where conceptually or theoretically you would have 100% of the population carry that gene drive. So how does it do it? If we go to the top part of this slide, we in the laboratory insert a transgene which encodes for these genome editing tools into 1 copy of the gene in the animal. As soon as we've done that, this copy activates and copies itself into the other copy of the gene. And thereby when the animal, be it female or male, that has gene drive, mates with a non-gene drive animal, all the gametes, sperm or egg, carry the gene drive. And as soon as that fertilization happens, the copying happens again, so that all the animals produced carry 2 copies of the gene drive. And in this way, it drives itself through the population. Next slide, please. So what can we do with such a tool? Well, we can drive any trait through the target population. Going back to the pigs we described at the start, if we applied a gene drive strategy to the target gene for PRRS resistance, which is CD163, we could drive that through a pig population. We could drive any trait, at least conceptually, any trait through a population. Not only that, but we would only function in that species. It's only possible in species that have sexual reproduction, and it is specific for that species. You could make gene drive specific for a subgroup within a species. I don't know if squirrels have breeds. I'm not sure they do, but let's say Aberdeen is an island, and the grey squirrels in there may have diverged just slightly genetically from other greys in the UK. We could conceptually design a gene drive only for those greys in Aberdeen. So it's an incredibly specific tool. It relies on the DNA sequence that this gene drive targets. So we could drive any. So in discussion with ESI, we feel that this tool could be used to drive infertility, female or male infertility. Quicker if it's female infertility, just because boys get around more than girls. That's all. But we could do either. The project is supported by ESI, and we've had some initial funding from them, which we're extremely grateful to. And this has allowed us to model what would happen if gene drive was deployed in one scenario of grey squirrels. And I would also, I'll come back to this right at the end, what makes this really attractive to, obviously, this is my day job. I'm passionate about genome editing and gene genetic engineering, and I could talk all day about it. But what appeals to me in the context of challenges like pests like the grey squirrel is that this is humane. There's no poisoning, there's no trapping, there's no killing. Animals don't die. They're just, some of them are infertile. Next slide, please, Fiona. So this conceptually, and I won't go into too much detail here, but I'm happy to discuss later. There's conceptually two ways we could do this for female infertility. We could have a gene drive, which we excitingly call the X shredder, which we could use to target the X chromosome and destroy it, and thereby any gametes produced by the animals would not have functional X chromosomes. There's a challenge to that in that we have to express the gene, make the gene drive active at the right time. The homing gene drive, conceptually simpler in that it targets a gene we know that's involved in fertility and activates it. Again, the diagram in the middle here shows that the copying event happens, destroying the activity of the fertility gene. And in this way, we would drive infertility through the population. Why insects? Well, the technology has been developed for mosquitoes. There's considerable efforts here in the UK, but also in the US and elsewhere in the world. The interest in mammalian species, rodents, squirrels, is relatively new. And there are both academic and commercial companies working on gene drive for controlling mosquito populations with the goal of reducing the likelihood that those mosquitoes transfer horrible diseases to ourselves. Next slide, please, Fiona. So this is the modeling that we did with the help of ESI's support. And I won't go through the whole process. We published this, and there's a paper in the scientific press if you want the details. But we modeled two population sizes, 3,000 and 30,000. We had the help of Craig Shuttleworth, who some of you will know, to give us sort of insight into squirrel population and dynamics. And we modeled the release of 100 gene drive squirrels, assuming a 1-year breeding cycle. And you can see that with the smaller population size for that release number, we were getting significant reduction in populations after 10 years, 12 years, and we're effectively eradicating animals by approaching 20 years. This is our first model. We've not done any other models yet, and there'll be ways of tweaking and increasing the efficacy of this, but I'm happy to touch on that in questions. Next slide, please. This is a platform technology. As I said, it's been developed for insects, for mosquitoes. It could be applied to a whole range of invasive pests. The requirement is that you have sex reproduction, that you know the DNA sequence, and that's very easy these days to do that. And you probably want a reasonable reproductive life cycle. If the animal takes 20 years to reproduce, gene drive is going to take an awful long time to have its effect. As I said, it's humane, and we've had conversations in regards to rodents, but we've also had conversations about the signal crayfish. Next slide, please. This is where I'm going to stop. It's my last slide. So, and I do appreciate it's been very high level, but gene drive can drive infertility through the target population. We can build in, and I'm happy to do this in questions as well, what's called a generation stop. You can have a situation where the gene drive is only active for a certain number of generations. One example is the daisy chain, although that's probably not what we'd use, but it's certainly a good exemplar. It's the genetic equivalent of the immunocontraception strategies, which are being trialed at the minute. And it can be dovetailed into whatever other control strategy you've got to give you that long-term locked-in control of numbers. As I said, it's applicable to pest species, specific and humane. We are currently looking at how to build a whole project around this, and I'm looking at a 5-year development and a 3-year testing, trialing phase. And then we'd expect to see the impact in the following 2 decades. And at that, I'll stop speaking to you and happy to take what I'm sure are going to be questions.
Thank you very much, Graham, and also Bruce, highlighting some of the issues we were all very well aware of, and also the science. We have had a selection of questions submitted prior to the webinar. Some of these have already been answered throughout the speakers' presentations. I've seen a couple come in the chat, but if I ask the first one that was submitted prior to the presentations, and this is from Stephanie Kaiser, our Confal's own Stephanie. If we were to successfully eradicate a local or regional grey squirrel population, what would be the impact on the ecosystem, in particular population dynamics, as the ecological niche previously occupied by the grey squirrel becomes free?
Can I answer that? Oh,
Yeah.
I have an opinion on that. I mean, at the moment, you've got an alien invasive species having a fairly negative footprint, both in terms of feeding and nesting for competition space. What they have done is successfully displaced our native species, the red squirrel. So if these gene edited squirrels were proven to be successful and proved to work and created that vacuum, and they were released next door to reds would just fill that space, and then they would get back to kind of how it was 100 years ago, where red squirrels were occupying that space.
Okay.
No, all positive as far as I'm concerned.
If I could add to that, we can certainly model that type of phenomenon. We can model, knowing the reproductive capability of both species, we can model how they might come back in, or indeed how they would interface with predators and so forth. So that is an important bit of modeling that could be done.
Okay. I'll ask one from the floor, and I can't see everybody's hands. If you are putting hands up, please don't, because I can't see you all. Put it into the chat. So a question from Jonathan Clive. Jonathan, do you want to unmute and ask your question, please?
Thank you. Can you hear me?
Yes,
Yeah.
you can.
I think part of it was answered by Professor Whitelaw, but it was I was wondering about sort of the 2 things I was asking about were was the outcome to completely annihilate or remove the species? Is that the outcome of it? But then you've talked about putting in a generational stop. So that the first part of my question was about what stops it, and the second was, is the outcome that we're looking for to completely remove the species, or are you looking to reduce numbers?
I'm sure Graham's got answers to that as well, but
yeah,
but conceptually we can control the generation. Now you can obviously then re-release another gene drive set of animals, and I think we would probably think about modeling what sequential release would look like. The possibility, given the ability to sequence so much nowadays, is that we could actually go into geographical areas. I talked about Aberdeen just because I know it's isolated, and there may be fortuitous genetic variation in some animals which will allow you to tailor the gene drive to specific subpopulations, or indeed the number you release in combination with the generation stop could allow you to perceive geographical areas that you could dramatically, if not eradicate, the numbers. But that all requires a lot more modeling to come up with what scenarios are possible.
Can I just add to that, Bruce? I mean, I think ESI is certainly clear that this is an alien invasive species. It is having dramatic and negative impacts on tree quality, tree growth, economics, environment in that space, and it has displaced the native red quite considerably through squirrel pox. It shouldn't be here. The long-term aim, we would like to see it removed from the British countryside. It will take a long time, and reds will take their place. In terms of, I've seen someone else has kind of said, well, what about the merits on pine marten? Pine marten spread, I entirely support ESI funded some of the early work in Ireland. The trouble with all current control mechanisms is that ultimately what you do is create a vacuum in your space. So whether you're a private estate or a public body or whoever controlling, you can control over your own land. You can't control your neighbour's squirrels. And you just get vacuums and backfill and backfill and backfill. And that just continues on, which is why it's like I mentioned the dripping tap earlier. The great thing about gene editing and pine martens are that they can reach other people's squirrels. And so you can then start to have a much bigger footprint in the landscape in terms of control of the population. The trouble with pine martens is that they won't go to all parts of the UK because they will only be happy in a limited landscape where there happens to be a very hefty tree cover. So I agree pine martens are a really helpful way, and when pine marten populations get up, they will start to make a difference, but they will only take us so far, and we will still get backfill where we are controlling or where we're in a landscape where there's no pine marten. So
thank you.
I could add just one little bit to that, just to leave any fears. I'm sure the pine marten would be quite happy to eat a gene-drive grey squirrel as much as a non-gene-drive squirrel. And if it did, there'd be no risk to that pine marten whatsoever. There's no possibility of the gene drive transferring to the predator.
A few questions have come in. We've had one from Heinz Trout prior to the webinar, and a couple on the same sort of area, which is public assurance, safety of technology and detrimental impacts on other species. Another one from Jonathan: what is the likelihood of mutations occurring that could render the gene drive mechanism becoming ineffective? And Stan Abbott as well. Stan, do you want to ask your question actually around sort of safety and public awareness?
I just thanks Caroline. Stan Abbott from the Woodland Trust. I just asked the general point: what would the reception be for what feels like the ultimate genocide weapon if applied to other species or more widely?
I will address all that in one way. So let me just address the easy one, which is, could it affect other species? The answer is no. The gene drive is absolutely specific for a DNA sequence, and you would design it such that that DNA sequence only existed in the greys, and we have enough sequence information to do that. The follow-on question in there was, what would biology kick in and create mutations and inactivate it? Yes, it would, and that will happen over time, and that will be a natural limiter on the gene drive activity. There would be a natural selection for those mutations once they appeared, and this goes back to having a generation stop. I think the combination of that and the ability to sequentially apply gene drives would negate that being a risk to how effective it would be. Concerning the public, there's I don't know if you I had to wake up at some very early hour this morning to go into the Today Go program, and at the end of it I got about 30 seconds of speech. I'm much more happy with this longer conversation. It wasn't about gene drive or squirrels, it was just about public discussions around genome technologies ahead of DEFRA's response to the recent consultation. My experience of interaction with the public and the surveys which many other groups, both academic and commercial, have done, is that the public want to realize the great potential of these tools and want to see what's possible. And when it comes to that, they want to see, top of the list, this is largely for farmed animals, welfare and sustainability. And to me, gene drives against grey squirrels, humane way of controlling numbers, is exactly what the public are after. So I think there is a huge dialogue to be done, and it is started. But I do believe there is a willingness in the public, and who is the public? The public is a very diverse beast. There'll always be people who align very closely with my own thinking, and there'll be people who are at the other end of the extreme. And and there's nothing we can do about that. But I do think there is a general trend within the public to see what is possible with genomic technologies, and with the top of the list is welfare and sustainability. And to me, gene drive for greys ticks those two boxes very heavily.
Can I just, oh, go on. I'll just add there that I think there's two levels of this. One is dealing with the public and the public view, particularly in relation to trees. Every time I've taken a group of people around a forest, might be a parish walk, whatever, I'll talk about grey squirrels and deer. And you tend to get, at the start of the walk, you tend to get this kind of slightly uninformed 50-50 view of whether or not controlling deer and or squirrels is a good idea. Actually, when you then show the damage physically to the people on the walk, without, every time I've done it, people come 99% your way. It's like, oh, I never knew squirrels damaged tree. I never knew deer did so much harm to the ground flora. My goodness, we'll stop feeding them on a bird table. So in terms of the loose public opinion about the negative impacts of the grey are easily won round if only we would all engage with everybody. That's my general answer. With regards to gene drive and genetic engineering, clearly there need to be caveats, and then we would have to go through protocols and testing and all the rest of it before this was ever started to release into the wider environment. I'm happy to go through that and need to go through that. You know, none of us wants this to go wrong. But if people have got a fundamental issue about genetics and gene editing, well, many of the vaccines were developed with gene editing. Europe had to adjust the rules on genetic modification in July last year to allow vaccine development in Europe for COVID. So this is a technology that's here. Yes, we need to use it carefully. Yes, we need to make sure that there are no negative impacts, but it is a technology that offers a great prospect, in my view.
I've just got one final comment, Karen, if I may. In one of my early slides, I indicated the Australian government have put some money towards gene drive for rodent control. This has been endorsed by PETA, you know, an organisation not known for endorsing applications in animals. So there is. I genuinely believe there's a change in mood when it comes to realising the needs that are out there.
Lots of questions coming in. I'm going to group them together while I can. So questions on trials and practicalities. So one from Toby Cook, very sensible question, and another much the same later on. How is gene drive administered? By injection or what?
You would.
And the second one, if I follow up with one as well about trials, how will trials areas be selected, and is there a process for landowners to be put forward for their land? So
I'll answer the first one, say a few things about the second one, and then pass maybe to Graham. So you would have somewhere you would have a breeding population of greys which you engineered to be gene drive, and you would release animals into the forests. When it comes to the trials, initially they would have to be containment trials in some large area. Perhaps the facility in York would be appropriate for that. And then after that, it would have to go into specific forests. I would be more than welcome to speak to anybody. In fact, when we have media announcements about this technology, I do get a lot of people writing to me saying, when you do trial it, use my forest, please. But Graham, you may have a specific comment about where and how.
Well, certainly. I mean, I just to echo those comments, you know, to get approval and then develop this is going to be a slow old process, and we want to walk carefully. But if in doing so, there would be containment trials. When it comes to an open trial, the ideal site, in my view, would be an island with just grey squirrels on, and see what happens. I don't know whether there is an island, a small island somewhere that has just grey squirrels. If there's anybody present on the call, let us know. In my mind, I think there are island populations on the mainland of grey squirrels. That are proximate to reds. So in my mind, that might be the best place to start, but we haven't actually set that fully out yet. We're going through this, working up the technology, working up the reagents. There's a long way to go before we get to the release of these things, if it's approved.
I'm going to ask a couple of people to ask a question. So Cheryl Durden, could you unmute yourself and ask your question, followed by Alex Barlow, please.
Hi everyone, thanks for putting me on. I was just wondering about some of the constraints facing gene drive, because it sounds so promising, and obviously lots of work has been initiated. So I was just wondering if there were any constraints at all.
So biologically, there's constraints in that no one has ever genetically engineered a squirrel, and we would have to obviously set up the capability to do that. Now, at Roslyn, we use similar strategies for mice, for rats, for chickens, for salmon, pigs, sheep, cattle, goats. We're just about to do goats. I'm relatively optimistic that we can adapt it for the squirrel, but we'd have to do that. We'd have to show that. Secondly, it is all very well done demonstrating something in an insect or indeed in mice, and we have a lot of information about fertility genes in mice, indeed in ourselves, and so forth. It's extremely likely that that would translate into the squirrel, but we'd probably have to try 2 or 3 targets just to make sure that it did what it wanted. This is still a bit of biology in there, which is never straightforward. So there's still much in the development phase which is going to challenge us, but I don't think any of that is unsurmountable given the knowledge we have in other species. Beyond that, we're going to get I think a helping hand from the insect community, who obviously can progress much faster than somebody working with mammals. And the data will come out of there, which will help us to refine our strategies for squirrels long before we have a squirrel on the ground.
Thanks, Bruce.
Can I just add, we are talking about a reasonable amount of money here to develop this, and that's a constraint, if you like, but we believe that there's, given proof of concept and all the rest of it, that's more than justified, which I'll come to at the end of the seminar.
Alex, could you ask your question next, please?
Yeah, my question is, it's going to make it far more complicated, but could you add some sort of gene coloring for the fur with the gene drive, so that you can actually identify that squirrels have gene drive? And then if you were doing culling, then you could differentially cull the ones that don't have the fur color, and that would speed up the decrease in the population.
So conceptually, you could. We routinely use a number of visual dyes. The one that you probably know of is one called the green fluorescent protein, which does exactly what it says on the tin. It makes an animal fluoresce green, and it's activated by blue light, and there's enough blue light and daylight for it to glow. And you can, even with a heavy fur covering, you can differentiate. So you could do that. Yes, you could. I've not heard anyone suggest that would be done, but yes, you could. Beyond that, you can obviously monitor the density of a gene drive within a given population. We would anticipate we could even work out that density on squirrel droppings and analyzing those, rather than actually analyzing animals themselves. But you might have to have some sentinel animals in there. But conceptually, you could make an animal green or blue or red. I always tempt my students by asking, can we make them tartan, since I'm Scottish? And we haven't found the gene that'll let us do that yet, but maybe tomorrow.
That isn't an aim of the project, but we'll go away and think about that one.
It's a great question, absolutely.
Yeah, great question.
It's a really good question, and I'm going to put it on my student list. Thank you very much, Alex.
Thanks.
We've got questions coming in. Esmond, were you wanting to ask a question then? You had your hand. I can just see you. I can't see many other people. You're on mute, Esmond. I can't. You're still on mute, Esmond. I'll pass on to. We've had questions coming in about this approach being applied to other pests. Midge deer and rats was one of them. You may have covered that in the presentation already. A good one, if you have covered it, I apologise, but Richard Hunter, who's our technical director at CON4, if not 100% effective, is there the possibility of genetic resistance developing?
There's the possibility of genetic resistance developing to anything that you apply to control any animal, and that goes back to my comment about putting in a sort of generation stop. If that's what was deemed to be the right approach, and indeed legislation for it, you would conceivably have a series of gene drives that you could deploy. The analogy that I'll give you is the coronavirus vaccine. Various companies have got a template now. If another variant comes out which is not protected by the current vaccines, we could make a new vaccine relatively quickly, certainly not the 12 months that took to get these ones, because the template is there. So we could have a series of gene drive templates that if escape, we call them escape mutants, but if animals were escaping from it, or you needed a secondary deployment of the gene drive, you could do that. So yes, it's possible. It's a constraint that affects everything that you try to do in this sort of way, but there are ways to mitigate that risk.
Perfect. So Esmond, I can see you now, and then if Richard Bellamy could come on after Esmond, please. Esmond, we can hear you now. No,
No, we can't.
we can't. Sorry, Esmond. Richard, please, Richard Bellamy. Richard, are you there? No. Gremlins. Pete Comley, you have asked an interesting question, or sort of a about process. Do you want to come on and ask your statement or question?
Yeah, it's a fairly simple question, which I must be failing to understand something here. But if you put a gene into a squirrel to make it infertile, how is that, and that you release 100 of those squirrels into the population, how are they ever going to spread that gene to other squirrels out there because they're infertile? Or am I missing something?
So no, you release the model we have is you'd release males, and those males would pass their gene drive on to the offspring. If they pass a Y chromosome at the same time as that gene drive, then that male is fertile. If they pass an X chromosome, where boys are XY, then the offspring will be infertile. So basically they produce fertile boys and infertile females. And it just keeps going until there's a population crash because there's not sufficient fertile females around. So they mate naturally, normally, and you'll get boys. This is for the homing design, not for the X shredder, but the homing. You'll get boys and girls born, but only the boys are fertile.
Get it.
The females will carry on living until they reach their death date, age-wise, but they will not reproduce.
And all the boys that get born carry the gene on, so they do their bit. Yeah,
correct.
I get it. Thank you.
Okay.
Very helpful. Thank you.
Right,
Sure.
I'm going to ask a couple of questions. So yeah, Richard Bellamy was asking, others asked about timescales. What sort of timescales are we envisaging?
So I think experimentally, we're talking about five years. We may be able to bring that forward depending on how the biology goes. We'd need a couple of years beyond that, at least, for the sort of contained trials, and then you would be able to release depending on regulatory positioning. Once you release the animals, that initial modeling we said is that you would start to see a population decline about a decade later, with extremely low levels, if not eradication, nearly a decade after that. We may, when we do further modeling, show that timescale can come forward, but we've just not done it yet. So it's not an immediate fix. And this goes back to a comment made earlier, that we see this as complementing current control mechanisms as the sort of final lock-in of reducing numbers and eradication.
Thank you. One more question from Jeremy Evans. If you're there, Jez, can you open your camera on, and then I'm going to revert to Graham Taylor to close.
Thanks, Caroline. That's great. Hi, Graham and all. I kind of answered my own question in my head a little bit. So I was just trying to think out the box a little bit, as you can see, but obviously it doesn't take away the fact that there's a habitat competition. But could it be complementary in any way, that alternative, or not? I guess it's too expensive as well.
It didn't come across too clearly, but I think it was about complementary approaches, and yes, absolutely. You can have a linking of a variety, and over a time scale, you can do that as well. I don't see this as an and/or type, sorry, an or type situation. It's an and type situation. You could deploy it alongside immunocontraceptive, for example.
Okay, sorry, Bruce, I was expecting you guys to see my text. Too much of an expectation. I'm sorry. In the text I've asked, can we turn the gene drive on for red squirrels so that they're immune against the pox? Right, okay, yes. So I've got a number of conversations in that regard, not least my colleague in the Morden, Colin. We could drive, same way we did the PRRS resistance in pigs, we could, but there's a problem. We have no idea what gene to target to give us squirrel pox resistance. If I could get a sizable pot of money to chase that, I would look at that, but we don't have that information. What we do have is candidate infertility targets.
Okay. Can I just add? Hi, Jeremy. Nice to hear your voice. I can't see you. But it doesn't get over the grey squirrel damage problem.
No, absolutely no. Yeah, quite right.
Yeah, but I see in the thread there's quite a lot of people saying, could we apply this to other things? So the RSPB is spending £6 million eradicating stoats. Yes, the answer, yes, it could be applied. The challenge is how to get, ultimately, if you're trying to eradicate a population, a non-native species, the IUCN the successful IUCN guidelines have all indicated that you need 3 vectors of control, i.e. you need gene editing and somebody still doing some trapping and some pine marten somewhere, or whatever the 3 different levels are to actually effect that control.
Sure. Yeah. Fascinating webinar. Thanks, guys.
Should I?
We have 2 minutes to go. So
okay. Fiona, can I ask you? Fiona, can I ask you to put up the last slide on my presentation, please? Is that possible? Is she still there? I can do it if she, yeah. Just while she's doing this, I sat last night musing on my 30 years in forestry, which is, I'm now in the last quarter of my working life, although as most foresters go on, don't they? But I sat back and looked at the stats from the published by the Forestry Commission. And these are the broadleaf planting figures in England over the last 30 years. Forestry Commission has felled and restocked 12,000 hectares, just under. It's about 400 hectares a year. Private sector has felled and restocked 37,000, nearly 38,000 hectares. It's just over 1,000, so it's about 1,200 hectares a year. Actually, in 2020, we pushed through 100,000 hectares of new planting over the last 30 years, which is an achievement. I then sat and thought, well, actually, how much does all this cost? And I put these are very back of the packet envelope calculations, but I used the average grant rate for that period and the average annual payments that are paid under had been paid under farm warden premium, farm woodland scheme, and latterly under countryside stewardship, and came up with these figures. To date, on average, about a quarter of a billion pounds is spent on planting those trees, and 440 million has been spent supporting their maintenance. That is not an inconsiderable amount of money. And we're just about to embark on the largest tree planting program that this nation has seen for the last 30, 40 years. I'd like you to answer the question in your own minds. Of that, what have you seen over the last 30 years, success or failure? I think if I'm looking at my own knowledge and experience, we've got probably only about 10% where we are absolutely on top of it, squirrels controlled, trees growing properly. At the other end, it's probably more like 10 to 20% of failure. Somewhere in between, there's the grey of part success and part failure. Now, I don't think it's acceptable that we're going to be spending another £690 million of public money to deliver the kind of forests that we want to achieve and will know we will fail to achieve in the absence of solving the grey squirrel problem. Therefore, I think £10 million is actually small beer compared with the gargantuan amount of money that we are just about to spend. These figures exclude the costs of administration, exclude supplements. So by the time you could probably, I wouldn't say double, I don't know what the ratios are. Somebody in the Forestry Commission could probably tell me, but they're not inconsiderable sums. And we know we've got a problem. So I think it's worth exploring.
Now, I know you've got a presentation coming up this afternoon with the administration. So I think a collective best of luck with that, because this really could be a game changer. And I would like to thank, firstly, everybody for joining the webinar. Thank you to Graham Taylor. Thank you to Bruce for superb presentations. And there will be an article in Forestry and Timber News for CONFOR members and non-members that will follow up on this webinar and perhaps expand on some of the issues and some of the points. But yeah, thank you so much, everybody. And I wish you a great weekend. Okay.